Bioprocess Online Live, multi-specific antibody edition. I'm Matt Pillar, editor at Bioprocess Online and host of the Business of Biotech podcast. I'd like to begin by thanking Cytiva for its sponsorship of today's discussion, which was originally going to be with two experts in the antibody development field, but unfortunately, Ian has been unable to join us this morning, so I am going mano a mano this morning. Not mano a mano, actually, because we're going to invite our audience to ask as many questions and help me interrogate Amir as possible. So we're going many a mano with Amir Goudarzi today. Dr. Amir Goudarzi is Senior Director of Bioprocess Technologies at Bayer. His expertise spans not just CGMP, bioprocess engineering, design, and manufacturing, but also CapEx and strategic road mapping, in addition to his influence on multiple of Bayer's antibody candidates. Dr. Goudarzi has more recently been involved in the development of Bayer's cell and gene therapy candidates, giving him a very holistic view, vantage point on the company's ATMP development efforts. He's particularly adept at leading global tech transfer of biologics from development labs to pilot to full commercial scale with a focus on industrialization of processes. So it makes him the perfect guy to go one-on-one with today. Dr. Goudarzi, welcome, and thank you very much for joining us.
Thanks a lot for inviting me, and thanks for the introduction.
Oh, it's our pleasure. It's our pleasure. Now, I want to reiterate something that I said at the top here. We are here for you, our audience members, and while I have plenty of questions for Dr. Goudarzi, your questions are just, if not more important than the ones I will ask. So to make sure you get the most out of your time with us, make sure that you're using that Q&A box and asking questions as you'd like. Dr. Goudarzi, I want to start with a conversation that you and I had not too long ago. I've had the pleasure of getting to know you over the past few weeks a little bit. And one of the, I think, common threads in our conversations about the development of multi-specific antibodies and monoclonal antibodies in general has been that speed and efficiency are kind of hand-in-hand primary goals of development efforts. So I know we're starting at a high level with this conversation, but just to sort of give us a level set on the scene, why is that so important right now?
I mean, always the time to market has been a very important factor in developing medicine. And I mean, a lot of effort is spent in small to large pharmaceuticals to understand what does it take to manufacture the product faster. Why is that? Is in order to actually streamline and reduce the cost of development from one perspective. From the other side, obviously, we're all doing this for a patient, right? If you're able actually to get this to the patient faster at a right price. So I think from the end in mind, end is the patient, that is one aspect that companies are competing actually to do that. Internal organization, which in a large organization is more complex, much more complex than a smaller company. And also the business landscape where we are in, right? So, I mean, unlike we have aspects around us that the environment that we're in that is changing, right? And that brings some level of uncertainty. So people would like to actually be able to understanding that, respond to that as fast as possible. So I guess these are some of the key aspects that we see, let's say, with regards to the acceleration of the development of antibodies or pharmaceuticals in general, right?
Yeah. Yeah, and it's sort of a segue into the topic of today's conversation around developing process efficiencies. I want to ask the flip side of that question. What challenges, particularly maybe starting with upstream, what challenges are standing in the way right now of gaining that speed, and how are we dealing with them? I'm thinking things like raw materials and tighter performance, that kind of thing.
Absolutely. I guess there are a couple of parameters looking at the upstream side that are impacting the, especially for multi-specific antibodies, the final performance or final process design that we have. Um, from one side is actually starts from the cell line, right? So we have diversity of the cell lines, and these different cell lines can introduce a variable in the performance aspects that we may not have seen before, right? That's actually on both ends. Some cells are very sensitive, some cells are not, and even I've seen it actually in companies that even when they have platform cell lines, they still, when we go to the new modalities, we still experience actually um, impacts due to them. Think about shear impact that we have, aspects that we have never sensed that for platform product, for let's say naked antibodies, but when we go to special types, we observe that. Another aspect is actually media and feed type diversity. So a lot of effort is actually spent in reduction of cost of goods, as well as intensifying or increasing productivity of these processes. That is to a good extent driven by the aspect of media and feed. So from one side, we like to simplify that the process to avoid having diversity of media and feeds. From the other side, we understand their impact. Um, so supply of them, requirements around them, preparation of them are aspects that are impacting, let's say, the multi-specific platform. And these issues are aspects that you may not see them at smaller scale, but the more that you go to the larger scale, you see them because at smaller scale, as opposed to larger one, the surface to volume ratio of the containers are different. And you observe aspects that your process is much less sensitive at smaller scale, but when you scale those processes up, the variability on the manufacturer side, media manufacturer, you sense them harder during your larger scale, commercial scale manufacturing. Often, actually, the variability of the cell lines and feeds leads to different matrix qualities that you have. So we're talking about different levels of HCPs, different level of DNAs, different cell density and viability of the cultures that we are ending up having. For these type of products, we often actually end up needing a smaller amount of product as opposed to the classical antibodies, and these are actually aspects that separation of the final product, it makes the separation also difficult, but also going down to the downstream, maybe if it's your second question, it will be actually to say that, well, requirements that we have, requirements are the same, but the methods of the separations, it's impacted, or the load on the downstream process impacted. So potentially for some of these processes, I've seen that a classic DSP process with a more known parameters is not immediately responding to the needs that we need to have there. And also hand in hand with that are basically product quality aspects. So depending on the type of the variants of the map that is actually being produced, depending on the process, you can also see actually a large number of half mirrors, dimers, or pieces of antibody floating around. So the in terms of high molecular weight or low molecular weight, you enter to an area that you haven't experienced before. So, and I think these all go hand in hand with the scale, right? Because at the end, is basically manufacturing it at a clinical/commercial scale. And I think the challenges with regards to scale up, scale characterization is something that is inherent to that. And maybe a fun part for us working in industry is that element of change. Well, it's a bit of a suffering and sweating at some point because you say, oh, I thought I know how it works, but but in the end, it's different. For example, please stop me, Matt, anytime you want, right?
You know, I'm going to be honest with you, Dr. Goudarzi. When I realized that Ian wasn't going to be able to join us, I didn't sweat a bit, because having gotten to know you a little bit, I knew that I'd be able to just ask you a question, wind you up, and watch you go. I am going to, and I want you to hold that thought just for a minute though. Remember where you were,
Sure.
because I'm going to interrupt you real quick, because we like to turn the tables on our audience and ask them a question or two during our presentation. So I'm going to ask Miles to throw up our first polling question. This question is for you folks in the audience. Simple. Are you primarily involved, you personally, you primarily involved in upstream process development, downstream process development, or both? Take a minute to respond to that question. We'll display the results here shortly. And I'll let Amir either pick up where he was leaving off, or I can hit him with another question.
I still remember it. I have a question
Okay.
still in my head.
Yeah, go ahead. Pick up right where you left off.
Cool. Yeah. So, I mean, as I said, I mean, on the upstream side, with regards to the changes that we deal with, as I said, they are fun at the same time. Makes you don't look very smart, and maybe that's the lesson. That's the lessons learned at the end that we can share. And it would be good if the experts online could comment on it. Is for example, there are processes. One of the ambitions of or wishes of people in this on the upstream side is actually to have no base addition to your culture. Often, actually, for this type of process, you a lot of times you do not need to have base additions, but it doesn't mean that you'd never need that. So and if you're actually used to a set of processes that you never need them, I've seen that actually people completely disconnect the pH and the base connection to the bioreactor, and yeah, it happens. And if you, I mean, I think my feedback to the colleagues is actually to myself, to remind myself and the colleagues, is actually always keep asking questions, actually why, right? Prior to running those processes, because it leads to catastrophes, right? And to large losses. So, again, another fun part is actually what brought us to here doesn't bring us further necessarily. We need to rethink, for example, with regards to the shear regime analysis across the scales to understand exactly and to mimic it what is happening at the development scale to the larger scale manufacturing. And otherwise, I've seen like a flat growth lines in the 2000 liter bioreactors, which we managed actually to overcome this after we realized that, well, shear profiling was not matching, and sales obviously sensed that and reacted.
Yeah,
so these are, I think, some of the important points that I thought could be good to share.